How do muscle trigger points form? We know the following events can cause muscle trigger points to form, but what physiological malfunctions cause them to form in response to these events? To illustrate this, we will use a simple model of a muscle fascicle that consists of only 6 muscle fibers innervated by 4 motor neurons. In addition to its motor neuron, each fiber has an arteriole that supplies it with blood for the oxygen and nutrients needed for energy essential to contraction. Finally, the fascicle is innervated by pain-sensing neurons called nociceptors that are sensitive to the absence of oxygen and the presence of ATP and inflammatory chemicals. The formation of a fiber trigger point starts with its motor neuron. Each fiber in a muscle is innervated by a single motor neuron that controls the fiber contraction. Nerve impulses in the motor neuron are conveyed to the fiber surface from the neuron's motor end plate. The end plate contacts the fiber in only a single location. The electrical impulse passes from the end plate over the fiber surface and causes the fiber to undergo a full cycle of contraction relaxation. Here is a close-up of a motor end plate. This view is simplified to show how the end plate stimulates the fiber contraction. In a previous scene, a simple model was used to describe how acetylcholine is released from the end plate, binds to an acetylcholine receptor to open a sodium channel, and produces an electrical impulse that spreads over the fiber surface and causes a contraction. To explain trigger point formation, the process is actually somewhat more complex. In reality, the fiber surface beneath the end plate has many receptor channels. Acetylcholine is actually released in units of around 10,000 molecules. Each acetylcholine unit activates a region of the fiber surface and produces a localized depolarization in that region called a miniature end plate potential. When motor neuron action potentials reach the motor end plate, the entire end plate is activated and 1 to 2 million acetylcholine molecules are released all at once. This mass release of acetylcholine produces widespread synchronous miniature end plate potentials that fuse into a grand end plate potential and produce a fiber depolarization that is then propagated over the length of the fiber surface, resulting in fiber contraction. However, acetylcholine can be released in lesser amounts than a maximum release. End plates can spontaneously leak acetylcholine molecules, and individual acetylcholine receptor channels are activated and form local depolarizations that are detected as spontaneous background end plate activity. It is also normal for motor end plates to release entire units of acetylcholine spontaneously and generate individual miniature end plate potentials. These individual miniature end plate potentials are too uncoordinated to form the grand end plate potential needed for a fiber contraction. However, when a muscle is damaged by trauma, exposed to infections, or undergoes prolonged tension by repetitive or persistent contraction, it can become inflamed, and the motor end plates become hyperirritable. Hyperirritable end plates show recurrent bursts of acetylcholine units that produce numerous uncoordinated miniature end plate potentials over the surface of the muscle fiber immediately below the end plate. Although abundant, these miniature end plate potentials are erratic and scattered and remain below the synchronized depolarization needed for a full end plate potential, and therefore fail to generate an action potential and a normal cycle of full contraction relaxation in the affected fiber. Nonetheless, the recurrent miniature end plate potentials appear to activate that sequence of molecular processes that cause a fiber contraction, but only in those sarcomeres that are immediately under the motor end plate. Since hyperirritability and its bursts of acetylcholine and the generation of miniature end plate potentials are all ongoing, the affected sarcomeres eventually undergo a maximum shortening and remain locked in a supercontracted state. The persistent unsynchronized bursts of neurotransmitter and their resulting weak but constant miniature end plate potentials result in a series of sarcomeres that accumulate into a hypercontracted knot in the length of the fiber under the end plate. Let us see this from the perspective of an intact muscle fascicle. Based upon our previous close-up, the fiber surface is being exposed to uncoordinated bursts of acetylcholine by the hyperactive end plate, and in response, it is generating multiple miniature end plate potentials. This causes the sarcomeres under the end plate to undergo maximal shortening, but with no relaxation. As the initial sarcomeres contract, neighboring sarcomeres are dragged under the end plate and stimulated to contract. Sarcomeres do not change their volume, so that sarcomeres under the end plate that are stimulated to shorten become wider, but adjacent sarcomeres outside the end plate that are not stimulated to shorten become stretched and thinner. This results in a knot forming in the fiber in the region under the end plate. Let us now explain how the miniature end plate potentials cause sarcomere hypercontraction.